Okay, so I'm going to explain to you how a vaccine that's being developed works. And specifically, this is the chadox 1 vaccine against SARS cov2 that's being developed by Oxford University. So first of all, let me explain that chadox is actually sort of a acronym kind of thing. So the CH of chadox is chimp, the AD is adenovirus, and OX is for Oxford University, so chadox. So what we're going to be using is a chimp adenovirus as the vector to shuttle the DNA that contains the gene for the spike protein into human cells, so the human cells can then secrete the spike protein so that you can make antibodies against it. So the reason why we can't use human adenovirus as a vector is that the human adenoviruses are fairly common cold virus, and most of us have already had that infection. So we would produce antibodies right away and eliminate or neutralize the vaccine, which would just mean it wouldn't work. So they're using a chimp adenovirus because we don't have prior immunogenicity to that virus. So adenoviruses are commonly used as vectors. So a vector is really just a way to get something in, like a Trojan horse, or like how rats are vectors for disease. They're a way of getting something in. So the adenovirus is a vector for DNA. So the adenovirus uses a receptor on cells called the CAR receptor. So I've just put a little CAR here so you can remember. It has a very, I don't know how to say this virus that starts with C, but it's a really hard to say acronym. So the CAR positive cell has a receptor that matches the adenovirus. And when they match, you get receptor-mediated endocytosis. So I'm going to remove the CAR for a second because it's hard to get this to go in. And you'll engulf the virus with the cell membrane. There we go. And then the DNA will eventually get inserted into the nucleus of the cell. There's the nucleus here. I'll move the nucleus. So the DNA of the adenovirus will go into the nucleus of the cell, and then transcription and translation can occur, which I will show you how that works. Okay, so I'm going to move the camera over a bit. Okay, so here's the cells. This is the person who got the vaccine. Now they have a piece of the adenovirus, the recombinant adenovirus's DNA inside the nucleus, and the promoter on the gene for the spike protein is a very hot promoter, so it's going to be transcribed really quickly. And then also engineered into this DNA is a signal peptide sequence that is going to cause the spike protein to be secreted. So right away, the nucleus is going to make the mRNA here, and the mRNA will have the signal peptide sequence, so that when the ribosome goes and translates this, the signal peptide will cause the ribosome to dock onto the ER, and the protein is going to be secreted into the ER. And so these are my models for spike protein that'll be produced in the ER. Oops. And then that will form a vesicle. And then the vesicle will then fuse with the cell membrane. And then you'll have the spike protein secreted out into the body. It's because the cells that get some of the cells that get infected by the adenovirus are endothelial cells, and so they could be secreted into the blood, apparently. Okay, so once you get the spike protein in the blood, then the regular immune response can occur because this is a foreign protein. So you're going to have dendritic cells and macrophages engulfing this, presenting the antigen to the T cells, or at least the T cells that can recognize that antigen, and then they're going to activate B cells, and then the B cells are going to produce antibodies, and then the antibodies will be able to bind to the spike protein. And of course, we don't want the antibodies to just bind to the spike protein that is produced by this vaccine, but later, when and if this person is actually infected by SARS-CoV-2, here I have my little model for SARS-CoV-2, this being like the spike protein, then these antibodies would bind and neutralize the virus. So that's how that vaccine works. And so I just wanted to show you really quickly how that is engineered. And so it's called recombinering, which is just genetic engineering, honestly. It's kind of cute. Kind of sounds like what Disney people do. So here is the adenovirus genome. There's a whole bunch of genes in the normal adenovirus genome, one of them being E1, which is necessary for DNA replication of the viral DNA. So we would remove that because we don't want the virus to actually replicate in the cell. We just want it to act as a vector to bring in the DNA that we want. And then we're going to take this E4 gene and insert inside the E4 gene. So we're going to interrupt this sequence. And that's where we're going to put this engineered gene of choice. So in this case, we have the gene for the spike protein. So that's going to produce the spike protein, which binds to, of course, our ACE2 receptors. You have the TPA signal peptide. That's so TPA is normally a protein that is secreted. So the signal peptide for TPA would cause this signal peptide sequence, DNA sequence, will cause a signal peptide to be produced, which will then cause the spike protein to be secreted. And then we want to have a hot promoter that's on all the time so that this actually gets expressed. So that's it.